Glycolysis, gluconeogenesis, and the role of aldolase gene expression by Hyla Torres. Glycolysis is the metabolic process where we are bringing in a 6-carbon molecule known as glucose in from the blood into a cell by breaking it down with various enzymes. In the process, it will release energy and furthermore pyruvic acid. It is both for anaerobic and aerobic respiration at the cellular level. I liked this image because it shows the variance and possible outcomes, which is always a good topic of exploration in biochemistry. Let's dig in and review the pathway for glycolysis. All right, so we've got a lot going on the screen, but we do have about 10 steps of oxidation and conversion in glycolysis. And I really liked this image here because it does give us a good picture of what is going on, getting us through the steps. So bear with me, try to follow along here with this image. The question to ask is, how does glucose enter the cell? That's what we're wondering here with this topic of glycolysis. Remember, glucose is a monosaccharide, which means it's a sugar. It comes into our body from our diet to provide our body with energy. It is water soluble, so it cannot passively pass the cell membrane via diffusion, so it must enter another way. The GLUT transporter is bidirectional, and there are various types of GLUT transporters. And to prevent glucose from exiting is by adding a special molecule here in the process. Isomerization occurs here to add a phosphate group to the glucose. Then catalyzation occurs by phosphohexoisomerase. So now we're going to dig into these steps. The initial step, starting with number 1 over here, is when D-glucose is converted into what is called glucose 6-phosphate. The enzyme responsible in this step is hexokinase. You can see that labeled right here. The next part is when the glucose 6-phosphate is rearranged into fructose 6-phosphate by an isomerase. Right here, this is what we've got. Then the phosphofructokinase uses a cofactor of magnesium to convert it into a fructose 1,6 biphosphate. So 2 sugars, which are isomers. Next, we have tricephophosphate, an enzyme quickly converting DHAP and GAP. The glyceraldehyde in this step is now being removed, so as you can see here, to be later used in the next steps of the glycolysis process. Glyceraldehyde 3-phosphate dehydrogenase will contribute a phosphate and in turn will produce 1,3-biphosphogycerate. Phosphoglycerate kinase transfers a phosphate group from 1,3-biphosphoglycerate from the given ADP to transform into ATP and 3-phosphoglycerate. An enzyme called phosphoglyceromutase relocates. So we got a little rearrangement going on. An enzyme, analase, will now take away a single water molecule from the 2 phosphoglycerates and will form phosphoenol pyruvic acid, or what is commonly known best as PEP. Finally, pyruvate kinase enzymes transfer a P from the PEP to ADP and pyruvic acid is formed, thus concluding our chart here. So hopefully this image gave a good visual of what's going on. Better than just hearing it is to visually see where these are going, where things are moving around, because there is a lot going on in the process of glycolysis. Now, we have another image here that I thought was important to show. I included this image as well for a few reasons. I think it's fantastic to not only look at one image to depict the pathway, but to look at multiple so no points are missed for the overall message. One can appreciate how, compared to the previous image shown, this is a more simplified version that still brings us to our outcome. So I think this one's a really good one if you're wanting to, if the previous one, there's obviously a lot going on here. I think it is a good one to reference when you are trying to visualize it. Once you have looked at it a few times, it's a little bit easier to move on to something more simplified like this with just the main points at hand. Of course, the arrows are very helpful. Now we're going to move on to gluconeogenesis. So please excuse this image here. I know that this image is a little bit, it's a little bit condensed, but that is because we've, again, got a lot going on here. So I'm going to go ahead and break it down. So we've got gluconeogenesis, and that is basically, when we break that word down, it literally means glucose. So when we're looking at gluco, that means glucose. Neo meaning new, and genesis meaning formation. So we've got gluconeogenesis is forming new glucose in the body system. This happens within the kidneys, specifically in the proximal convoluted tubule and within the liver of the body. This happens from non-carbohydrate sources like amino acids, lactic acid, and glycerol. It occurs primarily when blood glucose levels are low, so in the cases of hypoglycemia. Another reason this occurs is to support the brain. We know the brain relies primarily on glucose to function. Long-term fasting in an organism would cause the brain to rely on ketone bodies if need be. So with these two reasons, our blood glucose levels and brain fuel, we can see the importance of gluconeogenesis. In the pathway for gluconeogenesis, it's kind of the opposite of glycolysis in some ways, and we're going to talk more about that in some of the slides to come. So if that helps you kind of think of it to start off with, you can, but we're going to dig into whether that's really true or not. But in glycolysis, there are many reversible pathways. Some are not, and in this case, you have to go around that. So one thing I did really like about this image, even though it's got so much going on, you can see that there are a few arrows that are going both ways. So this is showing the reversible the possibility of the reversible reactions. And then there are some arrows that are only going one way. So it is important to notate the differences here. So I'm going to go over what those mean, just because it can be a little bit confusing, especially given what I just said. If you need to think about glycolysis being the opposite of gluconeogenesis, just hold that thought tight. And I want you to really think about that as we're digging into this. An example, so let's talk about lactic acid. Lactic acid coming from the muscles, maybe you're doing some type of strength training exercise. Lactic acid enters the cell and can later be converted into pyruvate. Pyruvate cannot go back in the pathway to PEP, so it needs to find a workaround. So you can see here, we've got our PEP, pyruvate, got to get a workaround. So that workaround is going into the mitochondria, and in the mitochondria, the pyruvate converts into acetyl-CoA. Another option is to convert into oxaloacetate. It does this by adding some form of carbon dioxide into the mix here. This is carboxylation. This is when the pyruvate carboxylase. This is a reversible step. A molecule named malate in this mix is pushed out of the mitochondria, and at that time, another enzyme helps convert it back to oxaloacetate. This is the true part of the workaround we just mentioned, because now we see the oxaloacetate convert to PEP by removing a CO2. I wish I was somebody who's really good at creating images or videos to show you how this can happen, but you're just going to have to, I guess, listen at this point. But that's essentially what's going on. This is the true part of that workaround. So, and at this time, the mechanism adds a phosphate in to get it to the PEP. This is called PepsiK or phosphoenol pyruvate carboxykinase. To stimulate this, by adding the phosphate, removing a carbon, then converting to PEP. From here, PEP can go back up the pathway until fructose 1,6-biphosphate comes in to help. Now, fructose 1,6-biphosphate can come in and trigger the opposite reaction by cutting off the carbon, a phosphate. Then a phosphate is going to release from this. Then you can work your way up the pathway a bit more. The glucose 6-phosphate moves to the smooth endoplasmic reticulum where an enzyme is inside. So once inside, the phosphate is ripped off the 6 carbon of glucose. The glucose will then exit through a glucose transporter and out the bloodstream. So remember, the initial reason for this was because the blood glucose levels were low. So that's what we're trying to bring it back to. We're trying to get that glucose back into the bloodstream so we can get those levels back up. This process helps return some of that glucose to the blood to help further bring them back to a healthy level. So that's what we've got going on with our brief overview of the gluconeogenesis. It's kind of hard to keep it brief because there are so many things that you need to acknowledge as part of this process. So hopefully that was a good simplified version for you. Obviously lots to dig into there. Another part important, we've been talking about glucose, so here's a couple images that you can see of glucose. Now normally in glucose entering the cell, glucose comes in via some type of GLUT transporter. So like a GLUT2 transporter for the liver or GLUT3 for the kidneys. When glucose enters the cell, it gets phosphorylated. It turns into glucose 6-phosphate. Then fructose 6-phosphate, then fructose 1,6-phosphate, and then it's split into glyceraldehyde 3-phosphate. Another component of that is dihydroxyacetone phosphate, or DHAP, and DHAP cannot move on, but it can be converted into glyceraldehyde 3-phosphate, which in turn can move on. Then the glyceraldehyde 3-phosphate is then converted into 1,3-BPG, 3-phosphoglycerate, 2-phosphoglycerate, and then to phosphoenol pyruvate. Finally, at this step, it is pyruvate. Now, remember earlier we were talking about glycolysis and gluconeogenesis being the inverse of each other. So let's talk about that a little bit more. That's something I don't want to leave as a gray area. So you can see here we've got a few different arrows, and based off of what we just talked about, hopefully you can guess which is which with the red and the blue. Hopefully you can identify that. So a little quiz time for yourself. And again, this image is yet another way to appreciate the pathway of glycolysis. I just noticed there are some parts cut off here, so you can see this is pyruvate kinase and fructokinase 1 we've got right here, so sorry about that. So I did want to include this image to segue into the topic of gluconeogenesis. It is common for some to get confused about these pathways. Again, I didn't want to leave it a gray area. You will see some similarities here, but it is important to address the question that we just brought up. Is gluconeogenesis the reverse pathway of glycolysis? In short, the answer is no. Gluconeogenesis is noting the reversible steps of the glycolysis pathway. So hopefully that was a takeaway you got from our previous slide. There are steps that are reversible, and there are some that are irreversible. Gluconeogenesis really involves the backward reaction of glucose generation. Glycolysis, as we just discussed, was really a role in form in pyruvate. So yes, it can be a little tricky, But just take note that the variance occurs with 3 steps, which are the conversion of pyruvate into phosphoenol pyruvate, the converting of fructose 1,6 biphosphate into fructose 6-phosphate, and last is the conversion of glucose 6-phosphate to glucose. Now we're going to talk about something I haven't really had the chance to mention yet, and that is our friend aldolase. Aldolase is a large enzyme that is found in the cell. It's in the nucleus, in the cytoplasm, and it's involved in the conversion process of fructose 1,6 biphosphate into dihydroxyacetone phosphate. Remember when we spoke about those in the pathways of glycolysis? It was a part of the 4th step where a diversion occurs, splitting the glucose into 2 separate molecules into pyruvate molecules. Aldolase mainly contains the amino acids lysine, and in the mechanism of aldolase, fructose approaches and enters the enzyme, and once it locates the lysine, it will bind to it, it will hang on to it. It takes the hydrogen ion from the oxygen in the glutamic acid. The arginine will steal 1 hydrogen from lysine, which will in turn form the double bond of lysine and carbon. Lysine will then remove a hydrogen from oxygen, which in turn forms a double bound carbon-oxygen after breaking the carbon-to-carbon bond. It essentially forms a new bond here and breaks the carbon-nitrogen bonds as well. The product outcome, named glyceraldehyde 3-phosphate, is created and then has the ability to leave aldolase, the enzyme. At this point, reformation begins in the nitrogen-carbon double bond. The carbon-carbon double bond breaks and hydrogen is removed from the arginine. Then water comes into addition and will break one of the nitrogen-carbon double bonds, and in its place will add a carbon-oxygen bond. Next, the oxygen here will form a double bond of carbon. A broken bond of nitrogen-carbon occurs and the removal of hydrogen from oxygen takes place. Another product appears. Dihydroxyacetone phosphate is here and released. Hydrogens rearrange at the site. In the human body, a bounty of aldolase isoforms are naturally occurring, particularly with glycolysis and the synthesis of glyceraldehyde. There is already much known about the aldolase. However, scientists are finding out even more about the functions that it has and also what it's putting out. It has been noted for its unique interactions with various proteins of the human body system. 3 isoforms of aldolase are worth mentioning in the glycolysis process are aldoa, aldab, and aldoc. I've seen it in various ways. You can see it in word form like this. You can see it where it says aldo-A or whatever letter is at the end. It's fairly simple to remember because we've got 3 isoforms, A, B, and C. So not too tricky there to remember the names. 3 isoforms, as we just mentioned, the aldoa, aldab, and aldoc. They're quite similar in their structured format. Aldoa is related to the muscles of the body as well as the red blood cell isoforms. Aldab are in tissues and organs, and aldoc with enzymes. Again, it's worth mentioning its relation to cancer and tumor progression. If overactivated in the body, studies have shown that the signal pathways correlate with the cancer progression. So a little real-life application there. We can really see how it's coming to play in an unfortunate circumstance. So with the cancer progression, a signaling cascade may occur and various proteins do get involved. To benefit the overall well-being of the organism, the signaling pathways occur from a cell working to complete its tasks and functions. For example, a study by Capsey and other authors with colorectal cancer patients observed such signaling pathways. Axin, a tumor suppressor called APC, and GSK3 played a role here. The aldolase isoforms also took part. Overall, the study showed that the isomers of aldolase were playing a role in regulation to the signaling pathway called WNT, and even for possible new cancer-fighting targets. Let's dig a little bit more into aldab, or aldolase B. For aldolase B, it is often found transcribed within specific organs of the body, which are the kidneys, liver, and within the small intestines of the digestive system. This isoform is the only of the three aldolase isoforms that are expressed in distinguished hepatocytes in the system. In regard to the splitting of fructose 1-phosphate into dihydroxyacetone phosphate and glyceraldehyde, it helps initiate the catalyzation. This is where we see the similarities of its presence in both glycolysis and gluconeogenesis. When you hear someone is intolerant to something, you may think, oh, they're probably intolerant to dairy or gluten or whatever the trend is these days that people are finding intolerances to. But did you know there are actually some people with the hereditary fructose intolerances? It's pretty rare, but it can be fatal. Excuse me. It can even be fatal if resulting from an aldolase B deficiency. This rare genetic disease is called hereditary fructose intolerance, and some of the symptoms in young people are irritability. So in babies, if you're looking for the symptoms, we've got irritability, neonatal jaundice, intolerance to fruits or avoidance of sugary foods, and even sleepiness. It is caused when the aldolase B gene is altered and mutated. When this mutation occurs, the instructions for making this gene do not play out, hence causing the issues with proper digestion of these foods. The ability to metabolize fructose here is heavily impaired for the person and can further hurt the liver cells by causing an overload of toxic forms of fructose 1-phosphate to occur. This will eventually lead to the liver cells to die over a prolonged period of time if they're repeatedly exposed to it. One study showed how a hepatocyte culture aldolase B were liver cells. This led to a result of transcriptional activity of the HNF3 nuclear factors. The PAB element in this case will bind to a unique matter to HNF1 and will further inspire the activity to continue. The study proved that HNF1, HNF3, CEBP, and DBP, plus more, were all part of the specific tissue transcription of liver cells in the body. So just to wrap things up today, hopefully you've gotten a good sense of what we've got going on with not only glycolysis and gluconeogenesis, but what type of a role does aldolase play in these two processes. Hopefully get an idea with that real life application that we were talking about with the cancer, the tumor progressions, things like that, even with that hereditary disease. Obviously rare, it's not something seen all the time, but it gives you an idea of how important it is to acknowledge these pathways and understand them if you are in the medical field, so you can recognize them, so you can recognize these blood glucose levels and just kind of the system overall. Thank you.